At its core, every living species operates with a single, overarching biological imperative: to survive long enough to pass its genetic material on to the next generation. For humans, executing this imperative creates a massive logistical hurdle. Half of the required DNA has to be packaged into a highly mobile, self-contained payload, the sperm, transported entirely outside of the body and kept alive in a completely foreign, often hostile external environment. To solve this, the male reproductive system functions like a precise biological assembly line. It relies on internal climate control, precise chemical synthesis, and specialized mechanical pressure to build and launch this genetic package. To understand exactly how this machinery works, we have to follow the physical path of a single cell, from its microscopic construction on the factory floor to the exact split second it creates a new human life. This lateral cross-section shows the system's architecture, including the internal duct network, accessory glands, and external anatomy. At the base is the scrotum. By hanging outside the pelvic cavity, it keeps the testes cooler, which is essential for temperature-sensitive cell production. Inside those cooled testes, a process called spermatogenesis begins. Triggered by hormones, base germ cells rapidly divide inside tight coiled channels called seminiferous tubules. They eventually shape themselves into distinct teardrop structures. But right now, these newly formed cells are completely immobile. They get pushed out of the testes and into the epididymis, this dense, tightly coiled tube visible on this microscopic histology slide. Think of the epididymis as a biological holding chamber. Over the course of several days, the cells mature here, developing their distinct physical tails and the mechanical ability to swim on their own. By the time they leave this chamber, the sperm are physically complete, but they still lack the chemical fuel they need to survive the journey and the fluid medium they need to actually swim. During sexual climax, physical contractions force the mature sperm upward. They leave the scrotum and travel into the pelvic cavity through a long muscular duct called the vas deferens. The sperm pass 3 critical checkpoints. First, seminal vesicles secrete fructose for energy. Next, the prostate gland adds an alkaline fluid to neutralize acidic environments. Finally, the bulbourethral glands lubricate the duct pathway for a smooth exit. Combine the raw sperm cells with these 3 distinct glandular secretions, and you get a complete biological mixture, semen. The mechanical delivery system for this mixture is the penis. 3 internal, sponge-like cylindrical chambers engorge with blood, stiffening the tissue to provide the rigidity needed to forcefully launch the semen out through the urethra. Through this synchronized sequence of physical manufacturing and chemical blending, raw genetic data is effectively shielded, fueled, and primed for an entirely different environment. Once the payload is delivered into the female reproductive tract, millions of individual sperm face an immediate, massive anatomical marathon. They have to swim entirely under their own power to reach the fallopian tubes. Only a tiny fraction of the original millions survive the distance. Those that do finally encounter their target, the secondary oocyte, or egg. The scale difference here is immense. Surrounded by thousands of microscopic sperm, a single egg looks like a massive planetary body. To gain entry, a successful sperm executes an acrosomal reaction. Its head bursts open, releasing a payload of highly specialized enzymes that dissolve a tunnel straight through the egg's thick outer barrier. The exact millisecond that first sperm breaches the cell membrane, the egg locks down. It triggers a cortical reaction, instantly hardening its outer layer into an impenetrable shell. This blocks all other sperm, ensuring the correct amount of DNA. Inside the egg, a final hidden step called karyogamy occurs. The sperm drops its tail, leaving only its genetic core. The chromosomes of the male and female cells drift together, collide, and permanently fuse. At this precise moment, a zygote is formed. The male assembly line has completely fulfilled its purpose, successfully carrying half of a human blueprint across the void to spark an entirely new life.